Welcome to my lecture on International Regulatory Submissions. Having covered the European Marketing Authorization Application (MAA) and the centralized procedure, we now turn our attention to Japan's New Drug Application (JNDA) process and briefly survey other major regulatory markets. Japan represents the third largest pharmaceutical market globally, following the United States and the European Union combined. Understanding the Japanese regulatory framework is essential for any regulatory affairs professional working in global drug development. The Pharmaceuticals and Medical Devices Agency (PMDA) serves as the primary regulatory body, though the approval process involves multiple governmental entities working in concert. The Japanese regulatory system has unique characteristics rooted in its historical development and cultural context. While Japan is a founding member of ICH and has adopted the Common Technical Document Format, the JNDA process incorporates specific requirements and review procedures that differ from both FDA and EMA approaches. This lecture will provide you with the foundational knowledge to understand these differences and their practical implications for global submission strategy. We will also examine regulatory processes in other significant markets including Canada, Brazil, and Australia, providing context for truly global pharmaceutical development programs. Today's lecture covers several interconnected topics that build upon your existing knowledge of the eCTD framework and regulatory submissions. First, we will examine the organizational structure of Japanese pharmaceutical regulation, including the roles of PMDA, the Ministry of Health, Labor and Welfare, MHLW, and the Pharmaceutical Affairs and Food Sanitation Council, PAFSC. Understanding how these entities interact is crucial for navigating the JNDA process effectively. Second, we will explore PMDA resources available to sponsors preparing JNDA submissions. PMDA has developed extensive English-language guidance and consultation services to support international sponsors, recognizing the global nature of modern drug development. Third, we will detail the JNDA process flow and timelines, highlighting key milestones and decision points. The iterative nature of the Japanese review process, with multiple formal and informal interactions between sponsors and regulators, represents a distinctive feature of this system. Fourth, we will survey new drug registration procedures in other non-EU ICH regions, specifically Health Canada, Anvisa, Brazil, and TGA, Australia. These markets present different regulatory frameworks and timelines that factor into global launch sequencing decisions. Finally, we will review key eCTD concepts to reinforce your understanding of the technical document format underlying all these regulatory submissions. Japan's pharmaceutical regulatory system operates within a complex governmental structure that differs significantly from the more consolidated approaches seen in the United States or European Union. The Ministry of Health, Labor and Welfare, MHLW, serves as the overarching governmental authority responsible for pharmaceutical policy, legislation, and final marketing authorization decisions. MHLW is a cabinet-level ministry with broad responsibilities extending far beyond pharmaceuticals, including labor policy, social welfare, and public health. Within this framework, the Pharmaceuticals and Medical Devices Agency, PMDA, functions as the scientific review body, conducting detailed assessment of application dossiers. Established in 2004 through the merger of several predecessor organizations, PMDA consolidated pharmaceutical review, post-market safety, and relief services into a single agency. The Pharmaceutical Affairs and Food Sanitation Council, PAFSC, provides expert advisory input at key stages of the review process. Council members include academic experts, clinicians, and other qualified professionals who deliberate on complex scientific questions and provide recommendations to MHLW. This tripartite structure means that sponsors must understand the distinct roles and interaction patterns among PMDA, PAFSC, and MHLW throughout the application lifecycle. Let us examine each of these organizations in greater detail to understand their specific responsibilities and how they contribute to the overall regulatory process. The Ministry of Health, Labor and Welfare, MHLW, holds ultimate authority for marketing authorization decisions. When PMDA completes its scientific review and PAFSC has provided advisory input, MHLW issues the final approval decision. MHLW also establishes pharmaceutical policy, develops regulations, and represents Japan in international regulatory harmonization efforts including ICH. PMDA's responsibilities encompass three main areas: review operations, safety operations, and relief services. The Review Division conducts scientific assessment of new drug applications, including evaluation of quality, safety, and efficacy data. PMDA also performs good laboratory practice, GLP, good clinical practice, GCP, and good manufacturing practice, GMP, inspections as part of the review process. PAFSC operates as an expert advisory body, convening to discuss scientific and medical aspects of applications at PMDA's request. Council deliberations inform MHLW's decision-making, though the Council's recommendations are advisory rather than binding. Understanding this division of responsibilities helps sponsors identify appropriate points of contact and communication channels throughout the regulatory process. This diagram illustrates the sequential flow of the JNDA review process, showing how applications move through the regulatory system from initial submission to final approval. The process begins when the sponsor submits the JNDA to PMDA. Unlike the FDA or EMA systems where a single agency handles the complete process, the Japanese system involves handoffs between organizations at defined stages. PMDA conducts the primary scientific review, which includes document review, compliance inspections, and scientific assessment. During this phase, PMDA may conduct multiple rounds of questions and sponsor responses, often accompanied by face-to-face meetings to discuss technical issues. Following PMDA's review completion, the application moves to PAFSC for advisory deliberation. The Council considers PMDA's assessment findings and may raise additional questions or concerns. PAFSC's input is particularly important for novel therapies or applications with complex benefit-risk considerations. MHLW receives the combined input from PMDA review and PAFSC deliberation to make the final authorization decision. The Minister formally grants marketing approval upon satisfactory completion of this process. The entire sequence typically requires 1 to 2 years, though timelines vary based on product complexity and the efficiency of sponsor-regulator interactions. This slide provides additional detail on PMDA's organizational structure and the resources available to sponsors preparing JNDA submissions. PMDA has made substantial investments in English-language resources and services, recognizing that many sponsors submitting JNDAs are multinational companies with international development programs. The PMDA website provides extensive guidance documents, application templates, and procedural information in English. The link shown on this slide directs to PMDA's Review Services Overview page, which provides detailed information on application categories, fee schedules, and procedural requirements. I encourage you to explore this resource, as familiarity with PMDA's online materials is essential for regulatory professionals working with Japanese submissions. PMDA offers pre-submission consultation services that parallel scientific advice meetings offered by FDA and EMA. These consultations allow sponsors to discuss development strategies, clinical trial designs, and submission plans before committing to specific approaches. Engaging in pre-submission dialogue can significantly improve application quality and reduce review cycle times. PMDA also participates in international regulatory cooperation initiatives, including joint reviews with FDA and EMA for certain products. These collaborative arrangements can streamline the global development process for sponsors pursuing simultaneous submissions across major markets. This slide displays the logos and organizational relationships among the three key entities in Japanese pharmaceutical regulation: MHLW, PMDA, and PAFSC. The Ministry of Health, Labor and Welfare, MHLW, website, accessible at the URL shown, provides information on pharmaceutical policy, legislation, and ministerial announcements. While primarily in Japanese, selected content is available in English. The PMDA website offers the most comprehensive English language resources for international sponsors. The agency has progressively expanded its English content to support global pharmaceutical development. Key sections include guidance documents, application procedures, consultation services, and approved product databases. The Pharmaceutical Affairs and Food Sanitation Council, PAFSC, operates within the MHLW structure. While less accessible to international sponsors due to limited English documentation, understanding PAFSC's advisory role is important for appreciating how scientific recommendations inform final approval decisions. The URLs provided on this slide should be bookmarked for future reference. Regulatory affairs professionals working with Japanese submissions need ready access to these authoritative sources of procedural information and guidance. Note that Japan operates primarily in Japanese, and while regulatory submissions may include English language components, formal documents and approvals are typically in Japanese. This necessitates translation services for comprehensive understanding of approval conditions and labeling requirements. Japan's good practice requirements align with international standards while incorporating specific national elements. Understanding these compliance frameworks is essential for sponsors conducting development activities intended to support JNDA submissions. Good laboratory practice, GLP, governs the conduct of non-clinical safety studies. Japan's GLP regulations are harmonized with international standards through ICH, but PMDA conducts its own GLP inspections of facilities generating data for Japanese submissions. International GLP-compliant facilities are generally acceptable, though PMDA reserves the right to conduct inspections of foreign sites. Good clinical practice, GCP, requirements for clinical trials in Japan have traditionally necessitated Japanese patient enrollment for most new drug applications. This Japanese bridging study requirement stemmed from concerns about ethnic differences in drug response. While ICHE 5 guidelines on ethnic factors have allowed greater reliance on foreign clinical data, sponsors should still carefully evaluate whether Japanese clinical studies are needed for their specific product and indication. Good post-marketing study practice, GPSP, governs post-approval surveillance requirements, which are extensive in Japan. Re-examination and re-evaluation periods require sponsors to conduct post-marketing surveillance and report findings to PMDA. These requirements often exceed post-marketing commitments required in other markets. Compliance with all applicable good practice standards is verified through PMDA inspection as part of the JNDA review process. Let us walk through the JNDA review process in greater detail, understanding the specific activities and interactions that occur at each stage. The process initiates when the sponsor submits the complete JNDA dossier to PMDA. The submission must conform to eCTD format specifications with Japan-specific Module 1 requirements. Upon receipt, PMDA conducts a preliminary validation to confirm completeness before commencing substantive review. During the review phase, PMDA evaluates all aspects of the application including pharmaceutical quality, CMC, non-clinical pharmacology and toxicology, and clinical safety and efficacy. Review teams conduct detailed assessment of submitted data and documents, often generating multiple rounds of questions requiring sponsor response. PMDA's review includes compliance verification through GLP, GCP, and GMP inspections. These inspections may target both domestic and international facilities involved in the product's development and manufacturing. Inspection findings are integrated into the overall review assessment. Expert meetings, involving selected PAFSC members with relevant expertise, discuss key aspects of the application. These focused consultations allow in-depth scientific discussion of specific issues, informing the broader review conclusions. A general review conference synthesizes findings from document review, inspections, and expert consultations to form PMDA's overall assessment conclusions. Following completion of PMDA's review activities, the process transitions to the final approval phases involving PAFSC and MHLW. PAFSC is formally consulted on the application, reviewing PMDA's assessment findings and providing advisory input. The Council may request additional clarification or raise concerns that require sponsor response. This consultation ensures that expert external perspectives inform the final decision. MHLW works with PAFSC to compile the final report and recommendation. The iterative dialogue between MHLW and PAFSC ensures thorough consideration of all relevant factors before the approval decision is finalized. Upon satisfactory completion of this process, the sponsor receives the marketing authorization grant from the MHLW minister. This authorization permits commercial manufacturing and sale of the approved product in Japan, subject to any conditions or post-marketing requirements specified in the approval. Throughout this entire process, multiple meetings occur between sponsors in both PMDA and MHLW. These interactions may be formal consultations or informal technical discussions, depending on the nature of issues being addressed. The frequency and depth of these interactions often exceeds what sponsors experience with FDA or EMA, reflecting the collaborative approach characteristic of Japanese regulatory culture. Effective engagement in these meetings requires thorough preparation and responsiveness to regulatory questions and concerns. The overall timeline for JNDA review typically spans 1 to 2 years from submission to approval. This slide illustrates the major phases and their approximate durations. Several factors influence actual timelines, including product complexity and novelty, quality of the submission dossier, sponsor responsiveness to questions, and regulatory workload at PMDA. The review clock operates somewhat differently than FDA or EMA systems. While target review times exist, the iterative nature of the Japanese process with multiple question and answer cycles can extend timelines significantly. Sponsors should plan conservatively when projecting Japanese approval dates. PMDA has implemented expedited review pathways for certain product categories, including designated orphan drugs, products addressing serious conditions with unmet need, and products eligible for Sakigake designation, Japan's breakthrough therapy equivalent program. These expedited pathways can substantially reduce review timelines for qualifying products. For global development programs, the Japanese timeline often represents the critical path for worldwide launch. Many sponsors pursue strategies to compress the Japanese timeline, including pre-submission consultations, high-quality dossiers to minimize question cycles, and proactive regulatory engagement throughout the review process. Understanding these timeline dynamics is essential for regulatory strategy development and commercial launch planning. We now turn to examine new drug registration procedures in other significant regulatory markets, beginning with Health Canada. Health Canada oversees pharmaceutical regulation in Canada through the Health Products and Food Branch, HPFB. The Therapeutic Products Directorate, TPD, and Biologics and Genetic Therapies Directorate, BGTD, conduct scientific reviews of new drug submissions. Canada's New Drug Submission, NDS, process accepts applications in CTD format, reflecting Canada's participation in ICH harmonization efforts. The review process shares conceptual similarities with FDA and EMA approaches, though with Canadian-specific administrative requirements and review procedures. Standard review timelines range from 6 to 10 months for new drug submissions. Priority review is available for products addressing serious or life-threatening conditions, potentially reducing timelines to approximately 6 months. Canada's proximity to the United States and cultural similarities make it an attractive early launch market for many sponsors. The relatively streamlined process and manageable timeline allow Canadian launches to follow shortly after U.S. approval in many cases. Health Canada has implemented progressive licensing approaches that allow iterative development of evidence throughout a product's lifecycle. This framework aligns with international discussions on adaptive pathways and real-world evidence integration. Brazil represents an important emerging pharmaceutical market regulated by Anvisa, Agência Nacional de Vigilância Sanitária, the National Health Surveillance Agency. Anvisa was established in 1999 as an independent regulatory agency responsible for pharmaceuticals, medical devices, food safety, and other health products. The agency has progressively strengthened its regulatory capabilities and international engagement. New drug registration in Brazil requires submission of comprehensive dossiers including quality, non-clinical, and clinical data. While Brazil is not an ICH member, Anvisa accepts eCTD formatted submissions, facilitating use of globally developed dossiers. Review timelines typically range from 12 to 16 months for standard submissions, though delays can occur depending on agency workload and submission quality. Anvisa has implemented priority review mechanisms for certain product categories. Brazil's regulatory requirements include specific provisions for tropical diseases prevalent in the region, local clinical data considerations, and Portuguese language labeling requirements. Sponsors must factor these Brazil-specific elements into their global development strategies. The Brazilian market's size and growth potential make it an important component of global commercial strategies, despite the relatively longer regulatory timelines compared to ICH regions. Australia's Therapeutic Goods Administration, TGA, regulates pharmaceuticals, medical devices, and other therapeutic products in the Australian market. The TGA operates within the Department of Health, conducting scientific evaluation of applications and maintaining post-market surveillance programs. Australia's regulatory framework reflects its Commonwealth heritage while incorporating ICH harmonisation principles. New drug registration proceeds through the Australian Register of Therapeutic Goods, ARTG. Sponsors submit comprehensive dossiers in CTD format, which TGA evaluates for quality, safety, and efficacy. Standard evaluation timelines approximate 11 months, with priority review available for medicines addressing serious conditions. The TGA's evaluation process shares conceptual similarities with other ICH regulators, though Australian-specific requirements apply. Australia participates in international regulatory cooperation initiatives, including work-sharing arrangements with Health Canada, Switzerland's Swissmedic, and Singapore's HSA through the Access Consortium. These collaborative arrangements can streamline evaluation for sponsors pursuing approvals in multiple consortium markets. The provisional approval pathway allows conditional registration of promising medicines while additional data is collected, similar to conditional approval mechanisms in other jurisdictions. Australia's English-language regulatory environment and alignment with ICH standards make it relatively accessible for international sponsors, often serving as an early launch market in Asia-Pacific strategies. We now transition to a review of key eCTD concepts, reinforcing the technical foundation underlying all regulatory submissions we have discussed today. This review section serves multiple purposes. First, to consolidate your understanding of the CTD framework that enables global regulatory submissions. Second, to highlight the consistency of format requirements across different regulatory authorities, and third, to prepare you for more advanced discussions of specific module contents in subsequent lectures. The Electronic Common Technical Document represents one of the most significant achievements in international regulatory harmonization. By standardizing the format and organization of regulatory submissions, the eCTD has dramatically improved efficiency for both sponsors and regulators. Understanding the eCTD is not merely technical knowledge, it represents fundamental professional competency for regulatory affairs practitioners. Every major regulatory submission you encounter throughout your career will utilize this framework, whether for initial marketing applications, post-approval supplements, or periodic safety reports. As we review these concepts, consider how the standardized format facilitates the international regulatory processes we have examined today, from FDA NDAs to EMA MAAs to PMDA JNDAs and beyond. The CTD pyramid represents the hierarchical organization of the common technical document, with increasingly detailed information at each level. At the apex, Module 1 contains regional administrative information that varies by regulatory authority. This module includes application forms, proposed labeling, and other jurisdiction-specific documents. Module 1 is not harmonized and must be customized for each regulatory submission. Module 2 provides summaries and overviews of the detailed information contained in the lower modules. The Quality Overall Summary, 2.3, Non-Clinical Overview and Summaries, 2.4 and 2.6, and Clinical Overview and Summary, 2.5 and 2.7, synthesize the extensive data packages into digestible executive-level documents. Module 3 contains comprehensive pharmaceutical quality, chemistry, manufacturing, and controls or CMC information. This module documents drug substance and drug product characteristics, manufacturing processes, control strategies, and stability data. Module 4 presents non-clinical study reports including pharmacology, pharmacokinetics, and toxicology studies. These studies characterize the product's biological effects and safety profile in non-human test systems. Module 5 contains clinical study reports and related information, documenting the human experience with the investigational product. This module typically represents the largest portion of a new drug application. The pyramid structure ensures that reviewers can access appropriate levels of detail depending on their needs, from high-level summaries to granular study data. This slide emphasizes several key characteristics of the eCTD that are essential for proper understanding. First, the eCTD is a collection of documents, not a single monolithic file. A typical new drug application may contain thousands of individual documents organized within the CTD structure. Understanding this modular nature is essential for document management and submission preparation. Second, the eCTD defines a format or organizational structure, a roadmap for how to assemble and present regulatory information. The format specifies where different types of information should be placed within the document hierarchy, enabling consistent organization across all submissions. Third, the eCTD's purpose is harmonization, creating consistency in how pharmaceutical companies worldwide prepare and submit complex regulatory dossiers. Before CTD harmonization, sponsors had to reformulate submissions entirely for each regulatory authority, a tremendously inefficient process. The harmonization achieved through ICH means that core technical modules, 2 through 5, can be largely consistent across FDA, EMA, PMDA, and other ICH region submissions. Only Module 1 requires significant customization for each authority's administrative requirements. This harmonization represents tremendous value for global pharmaceutical development, enabling efficient preparation of submissions for multiple markets from a common technical foundation. The eCTD format emerged through ICH harmonization efforts over several decades, representing collaborative work among regulatory authorities and pharmaceutical industry representatives from the United States, European Union, and Japan. The ICH M4 Guideline Series establishes the organizational structure and content requirements for the CTD. M4 itself provides the overall framework, while companion guidelines address specific sections, M4Q for quality information, M4S for safety, non-clinical information, and M4E for efficacy, clinical information. The three main aspects of harmonization, quality, Q, efficacy, E, and safety, S, correspond to the fundamental dimensions of pharmaceutical evaluation. Every new drug application must demonstrate that the product can be manufactured consistently, quality, provides therapeutic benefit, efficacy, and has acceptable risks, safety. The CTD structure organizes information to address each of these dimensions systematically. Understanding that the eCTD is a format applied to specific application types is important. The most common application of CTD format is for new drug or biologic applications, NDAs in the United States, MAAs in Europe, and JNDAs in Japan. However, CTD format is also used for subsequent applications including supplements, variations, and periodic safety reports. The format continues to evolve through ICH updates, most recently with the transition to eCTD version 4.0 specifications. This slide displays the ICH logo and references the M4 guideline series that establishes the CTD framework. The M4 series comprises four main guidelines: M4, Organization of the CTD, M4Q, Quality, M4S, Safety, and M4E, Efficacy. Together, these documents provide comprehensive guidance on dossier structure and content expectations. ICH M4 has undergone several revisions since its initial adoption. The current version, M4, R4, incorporates lessons learned from implementation experience and aligns with evolving regulatory expectations. Staying current with ICH guideline revisions is essential for regulatory professionals. The ICH implementation status varies by region and guideline. While all ICH member regions have implemented the core CTD requirements, specific implementation details may differ. Checking current implementation status through ICH's website ensures compliance with applicable requirements. The visual representation on this slide illustrates how the M4 guidelines map to different modules of the CTD. Quality information, M4Q, informs both Module 2.3, Quality Overall Summary, and Module 3, Quality. Safety information, M4S, guides Module 2.4/2.6 and Module 4. Efficacy information, M4E, structures Module 2.5/2.7 and Module 5. Familiarity with these ICH guidelines is fundamental to regulatory affairs practice. I recommend downloading and studying the current versions of all M4 series guidelines. The four overarching ICH guidelines for eCTD content provide detailed granularity on the requirements for each module. M4, the overall eCTD guidance, establishes the general organizational structure, formatting requirements, and cross-referencing conventions. This guideline provides the framework within which the more specific quality, safety, and efficacy guidelines operate. M4Q, specific to pharmaceutical quality information, details the content requirements for Module 3, Quality, and the Quality Overall Summary in Module 2.3. This guideline addresses drug substance, drug product, manufacturing process, control strategy, and stability documentation. M4S, specific to non-clinical safety information, guides the content of Module 4, Non-Clinical Study Reports, and the non-clinical overview and written summary sections of Module 2, 2.4 and 2.6. This guideline addresses pharmacology, pharmacokinetics, and toxicology study reports. M4E, R2, specific to clinical efficacy information, establishes requirements for Module 5, Clinical Study Reports, and the clinical overview and summary sections of Module 2, 2.5 and 2.7. The R2 revision reflects updated expectations for clinical documentation. Together, these guidelines provide comprehensive direction for preparing regulatory dossiers. Regulatory affairs professionals must be thoroughly familiar with applicable sections when preparing or reviewing submissions. Module 1 contains regional administrative information that is not harmonized across ICH regions. Each regulatory authority specifies its own Module 1 requirements reflecting national laws, regulations, and administrative procedures. For FDA submissions, Module 1 includes forms such as Form FDA 1571, IND application, Form FDA 356H, application form, Form FDA 3674, certification of compliance, and the U.S. prescribing information formatted according to the physician labeling rule. For EMA submissions, Module 1 includes the EU application form, summary of product characteristics, SMPC, following the quality review of documents, QRD, template, package leaflet, labeling, and various administrative documents including expert statements. For PMDA submissions, Module 1 includes Japan-specific application forms, Japanese package insert, and other administrative documents required under Japanese pharmaceutical law. The non-harmonized nature of Module 1 means that sponsors must prepare distinct regional components for each market, even when Modules 2 to 5 remain largely consistent. This represents a significant workload in global submission preparation. Module 1 organization follows a standard numbering scheme, 1.1 through 1.14 in the current ICH specification, but the content within each section varies by region. Understanding these regional differences is essential for international regulatory affairs practice. Module 2 contains the summaries and overviews that provide high-level synthesis of the detailed information in Modules 3, 4, and 5. This slide shows the table of contents structure for Module 2 as specified in ICH M4, R4, and the FDA's Comprehensive Table of Contents. While ICH M4, R4, provides the harmonized structure, FDA has published its own Comprehensive Table of Contents, headings, and hierarchy document that includes minor variations. These differences are largely organizational rather than substantive, but regulatory professionals must be aware of them when preparing submissions for different authorities. Module 2.1 contains the table of contents for the entire CTD submission. Module 2.2 provides the introduction, offering a brief overview of the application. Module 2.3 contains the Quality Overall Summary, QOS, synthesizing pharmaceutical quality information from Module 3. The QOS provides reviewers with a comprehensive yet concise summary of CMC information. Module 2.4 presents the Non-Clinical Overview, a critical evaluation of non-clinical findings. Module 2.6 contains the non-clinical written summaries, organized by discipline, pharmacology, pharmacokinetics, toxicology. Module 2.5 contains the Clinical Overview, a critical assessment of clinical findings. Module 2.7 presents the Clinical Summary, with subsections covering biopharmaceutics, pharmacokinetics, efficacy, and safety. The Module 2 documents are often the first materials reviewers examine, making their quality critically important. The organization of Module 2 summaries follows the guidance in the three discipline-specific ICH guidelines, M4Q for quality, M4S for safety, and M4E for efficacy. M4Q guides the Quality Overall Summary, Section 2.3, and corresponds to the detailed quality information in Module 3. The QOS follows a defined structure addressing drug substance, drug product, and supporting quality information. M4S guides the Non-Clinical Overview, Section 2.4, and Non-Clinical Written Summary, Section 2.6, corresponding to the study reports in Module 4. These summaries synthesize pharmacology, pharmacokinetics, and toxicology findings. M4E guides the Clinical Overview, Section 2.5, and Clinical Summary, Section 2.7, corresponding to the clinical documentation in Module 5. These summaries present the human experience with the product in an integrated fashion. The implementation status displayed on this slide shows that M4Q has been implemented across all ICH regions. Similar implementation has occurred for M4S and M4E, though specific regional variations may apply. When preparing Module 2 documents, authors must carefully follow the applicable ICH guidelines while also considering any region-specific expectations. The quality of Module 2 documents significantly influences reviewer assessment and can affect review timelines. This slide presents information related to Module 3 quality. Module 3 contains comprehensive pharmaceutical quality, CMC, information documenting everything necessary to ensure consistent manufacturing of a high-quality product. The Module 3 structure addresses both drug substance, active pharmaceutical ingredient or API, and drug product, finished dosage form. Each is documented through a parallel structure covering general information, manufacture, characterization, control of materials, control strategy, reference standards, container closure, and stability. Drug substance sections 3.2.s, document the API including nomenclature, structure, physical properties, manufacturing process, process controls, and specifications. Analytical methods and validation data support the control strategy. Drug product sections 3.2.p document the finished formulation including composition, pharmaceutical development rationale, manufacturing process description, excipient controls, and finished product specifications. Stability data supports shelf life determinations. Regional information 3.2.r includes region-specific quality information that may be required by particular authorities. The depth and complexity of Module 3 documentation reflects the fundamental importance of pharmaceutical quality. Consistent manufacturing of quality product is the foundation upon which safety and efficacy evaluations rest. This slide continues the presentation of Module 3 quality information, likely addressing additional aspects of pharmaceutical development and manufacturing documentation. Module 3 documentation supports the CMC review that occurs in parallel with clinical review during regulatory evaluation. Quality reviewers assess whether the sponsor has adequately characterized the product and demonstrated capability for consistent manufacturing. Key areas of focus typically include synthetic route and process controls for drug substance, formulation development and optimization for drug product, analytical method development and validation, specification justification, stability program design and results, and manufacturing process validation approach. The quality overall summary in Module 2.3 synthesizes this detailed information for efficient reviewer access. However, reviewers frequently drill down into Module 3 for detailed assessment of specific quality aspects. For complex products such as biologics, Module 3 documentation is particularly extensive, addressing cell line development, manufacturing process characterization, and the comprehensive analytical characterization required for these products. Strong CMC documentation is essential for regulatory success. Quality deficiencies frequently result in application delays or refusal to file determinations. Investing in robust pharmaceutical development and thorough documentation pays dividends throughout the product lifecycle. This slide continues Module 3 quality documentation, likely addressing specific aspects such as stability studies, container closure systems, or regional quality requirements. Stability documentation represents a critical component of Module 3, supporting the proposed shelf life and storage conditions for the product. ICH Q1AE guidelines establish the framework for stability study design, and Module 3 must present stability data in accordance with these requirements. Container closure system documentation addresses the primary packaging components that directly contact the product. Compatibility studies, extractables and leachables assessments, and functional suitability data support the proposed container closure system. For products requiring special handling, such as biologics requiring cold chain storage, additional documentation addresses storage and distribution controls to maintain product quality throughout the supply chain. Regional quality requirements vary somewhat across regulatory authorities. While the core quality documentation is harmonized, specific regional expectations may necessitate additional information or alternative presentations for different submissions. The iterative nature of pharmaceutical development means that Module 3 documentation evolves throughout the product lifecycle. Post-approval changes require updated CMC documentation through supplement or variation submissions. Module 5 contains clinical study reports and related information documenting the human experience with the investigational product. This slide displays the table of contents structure for Module 5 as specified in ICH M4E and the FDA's Comprehensive Table of Contents. The Module 5 structure organizes clinical information logically, beginning with a tabular listing of all clinical studies, 5.2, followed by detailed clinical study reports and related information, 5.3. Section 5.3.1 contains biopharmaceutical studies including bioavailability, bioequivalence, and in vitro in vivo correlation studies. These studies characterize the product's pharmaceutical performance in humans. Section 5.3.2 presents studies using human biomaterials, including plasma protein binding studies and hepatic metabolism/drug interaction studies. These provide important pharmacokinetic characterization data. Section 5.3.3 contains human pharmacokinetic studies, organized by population, healthy subjects, patients, and factor type, intrinsic factors, extrinsic factors, population PK. This section documents the product's disposition in humans. The systematic organization facilitates reviewer access to specific study types while enabling comprehensive assessment of the clinical pharmacology program. This slide continues the Module 5 table of contents, addressing additional clinical study categories. Section 5.3.3.4 contains extrinsic factor pharmacokinetic studies examining the effects of external factors, such as food, concomitant medications, or smoking, on product pharmacokinetics. These studies inform dosing recommendations and drug interaction guidance. Section 5.3.3.5 presents population pharmacokinetic study reports, which characterize variability in drug exposure across patient populations and identify influential covariates. Section 5.3.4 contains human pharmacodynamic studies, examining the product's biological effects in humans. These may include biomarker studies, dose-response characterizations, and pharmacokinetic pharmacodynamic modeling. Section 5.3.5 presents the pivotal efficacy and safety studies, organized by study design. Controlled clinical studies pertinent to the claimed indication, 5.3.5.1, typically include the Phase 3 trials that provide the primary evidence of efficacy. Uncontrolled studies, 5.3.5.2, integrated analyses, 5.3.5.3, and other studies, 5.3.5.4, provide supporting evidence. Section 5.3.6 contains post-marketing experience reports when available from other markets. Section 5.3.7 provides case report forms and individual patient listings, allowing detailed review of individual patient data when needed. Section 5.4 contains literature references cited throughout the clinical documentation. This final slide addresses the relationship between Module 5, detailed clinical study reports, and the Module 2 clinical components, clinical overview and clinical summary. The clinical overview, Section 2.5, should be a relatively concise document of approximately 30 pages that provides a critical assessment of the clinical data package. As noted in ICHM 4E guidance, this document should use graphs and concise tables to facilitate understanding while avoiding repetition of material presented fully elsewhere. The clinical overview serves as an interpretive document, presenting the sponsor's integrated analysis and conclusions regarding the product's clinical profile. It should address the product's benefits in the target population, its risks and how they can be managed, the adequacy of the clinical program to support the proposed indication, and any outstanding questions or limitations. Cross-referencing from the clinical overview to detailed presentations in the clinical summary or Module 5 is encouraged. The overview need not repeat extensive detail available elsewhere but should guide reviewers to relevant information. The clinical summary, Section 2.7, provides more detailed summaries organized by topic: biopharmaceutics and associated analytical methods, clinical pharmacology studies, clinical efficacy, and clinical safety. These summaries are longer than the overview but still synthesize rather than reproduce study reports. Together, the Module 2 clinical documents and Module 5 study reports provide complementary levels of detail for regulatory assessment. Thank you for your attendance.